Good morning, my name is Francesco Pavone and I work at the European Laboratory of Nonlinear Spectroscopy, an infrastructure of the European Community, and to the University of Florence. Today I'm going to talk about skin photobiomodulation therapy. And I will concentrate my attention on a couple of topics. The first one is try to understand together the biochemical principle of photobiomodulation, and the second one will be to show you some examples of wound healing treatment with skin photobiomodulation. So how does the photobiomodulation works? So mainly what we call phototherapy is based on two effects. One is the fact that the photon absorbed by the tissue, they are going to interact with some chromophore, delivering some energy, increasing the temperature. And if we go to temperature more than 45 degrees, we induce apoptosis of cells, so the death of the cells. This is what is called photothermal phototherapy. On the other side, there is a pure photochemical effects, which is due to the fact that the photons are again interacting with chromophores and inducing a photochemical effects. But then we may have two classes of chromophores. The exogenous chromophores that we insert inside of the subject from the external. A typical example is the photodynamic therapy. And here, because the photons are interacting with the chromophores, they are creating some photochemical effects, which is healing or killing also the tissue. The other one is the interaction of photons with endogenous, naturally present chromophores. And again, here we have a photochemical effects whose components activate cell pathways which produce healing processes. Photobiomodulation therapy is commonly used to alleviate pain, to promote wound healing and tissue regeneration. And to do that, we can use different kind of light sources. We can use a simple white lamp, which has an emission of a black body emission that is very common, where we have mostly the light centered into a frequency which depends on the temperature. Then we have a more monochromatic light produced by an LED. And if we want to push even more the monochromacity of the light, we can move to the laser emission. So I have to say that the density of photons in frequency is much more higher with lasers than with LED, than with a lamp, with white light. But another, let's say, characteristics that we have with lasers is that lasers are collimated. So I'm able to concentrate the photons on a much, let's say, smaller spot than a LED, which is a much larger illumination. So in terms of brilliance, irradiance, and fluence, the lasers are winning. But the question is, do we really need lasers for photobiomodulation? Or does it work also with LED? Because the LED has the advantage of being much cheaper, and we may have a very high power LEDs for a few euros. The answer is yes, and it's very easy. Why? Because the main component of the photobiomodulation is the cytochrome C complex. The cytochrome C complex, if you look at the absorption spectra of the cytochrome C complex, you see that it has two main peaks of absorption, one around 420 nanometer and the other one around 600. But at 400 nanometer, the cytochrome absorbs about 10 times more than the red. So if I want to be more effective, it's better to use blue light than red light. The problem is that, as we will see, that the blue penetrate differently, but we are going to discuss about that. But let's concentrate first on the photochemical effects. What happens when the light invests as cells? Well, it invests all the compartment of the cells and invests also the mitochondria. The mitochondria has inside a very important component, which is the cytochrome. And when light interacts with the cytochrome and in principle with the heme complex, it delivers the nitrogen oxide, it bounces a molecular oxygen, but the nitrogen oxide, as you can see from this slide, is going to create signaling to produce components, metabolites that are going to change completely the metabolism and the activation process of cells. In parallel, we have also an influence on the ATP and ROS production because of this photochemical effect. And again, the ATP and the ROS are going to change the transcription factors to influence the action of the transcription factors, which is going to change completely the gene transcription. So we may conclude in a little bit provocative way the fact that the light is somehow resetting, reprogramming the metabolism, the cell signaling of cells. Now, photobiomodulation therapy is commonly used for several applications. We go from antimicrobial effects to immunomodulation effects, to the regulation of pigment disorder, to the inhibition of inflammation relief from pain, up to wound healing, where we observe promotion of regeneration and prevention of tissue death. And that's the subject we're going to talk now. In order to understand how effective is photobiomodulation, we have to consider a couple of things. First of all, how deep can I penetrate with light in a tissue? There are many two things, absorption and scattering. And that depends pretty much on the density of the tissue, the scattering, and the absorption on the matrix, chemical matrix, molecular composition of the tissue. And if we have a look, as you can see in this slide, to the absorption spectrum of oxy and deoxyhemoglobin, melanin and water, we see, of course, that changing the wavelength, we can go deeper or less deep. So, for example, in the blue, there is a much higher absorption of the oxy and deoxyhemoglobin, and so to go deeper, we have to move throughout to the near infrared. But if we move too much into the near infrared, again, we have water. So we have a sort of, let me call, optical window between in the visible where we can penetrate more. But the problem is that if we are moving from the blue to the red, the absorption capability of the cytochrome is reduced. So we have to find a compromise. We have to understand how those two effects are balancing themselves. And in order to do that, we realize a digital tissue phantom. What is a digital tissue phantom? It's a simulation of a piece of tissue. In this case, is the first layer of the tissue, as you can see here. We are displaying the simulation in the vertical, so going deeper into the cell. Okay. And you see that we are working in a first layer from 0 to 2 millimeters. You see that moving from the blue to the red, we penetrate much more because the scattering is reduced. But what about the absorption? So if we put all together absorption and scattering, and we simulate the tissue with a Monte Carlo simulation, we see here, for example, that we are able to study the effects of interaction of light with cytochrome. For example, in this tissue phantom, we inserted 3 inclusion of cytochromes at 3 different depths. The first one very superficial, 0.25 millimeter. The second one a little bit deeper, 0.75, and the third one a little bit deeper, 1.25. Now, if we are studying what is happening when we are illuminating with blue at 420 nanometer, with red at 660, and with near infrared at 810, we see something which is quite obvious, let me say. Blue is illuminating the first layer, for sure, okay? But it's also absorbed much more than the red and the near infrared, which is, of course, propagating on the surface, like the blue. And the fact that the cytochrome is absorbing much more, we see that the effect of photoactivation of cytochrome on the surface with blue is 30 times more than the red and 100 times more than the near infrared. Then you could say, okay, but we are interested also to go deeper. And everybody knows that red and near infrared is going deeper. Correct. This is true. In fact, if I look at this second simulation, I can study the photobioactivation of the first inclusion, the superficial one with blue, the second inclusion a little bit deeper with red because red is going deeper, and the third inclusion a little bit deeper, even more, with near infrared, because near infrared is going even deeper. Now, it's true that red and near infrared are going deeper, but it's also true that the cytochrome oxidase absorbs much less in the red and even less in the near infrared. So in that case, what is the results? Is that, okay, we are photoactivating cytochrome deeper with red and near infrared, but with near infrared, we are about 300 times less effective to photoactivate the cytochrome than the blue on the surface. And with the red, we are about 100 times less effective to photoactivate the cytochrome at medium depth than the blue on the surface. So in that case, blue works much better on the surface, penetrate less, but going deeper, the red and the near infrared absorbs much less the cytochrome oxidases. So the question now is, but we observe quite a good effect going deeper with the blue, and in the next slides I will show you the comparison between blue and red. How does the healing process going deeper if the photons cannot go deeper? Well, we found quite a reasonable answer here, because we used a real tissue phantoms, not a digital one, and we studied the activation of cytochrome, and in particular cytochrome oxidases, cytochrome B and cytochrome C, with blue light. And we see that effectively there is a big increase in the density, in the concentration of those pieces. But the nice thing is that when we stop irradiation with light, we see an increase of that concentration with time. So there is a sort, let me say, of memory effects. And we would like to understand that. And from preliminary measurement, we saw that this is a very interesting experiment. That if we take some fibroblasts and we irradiate with blue, then we remove the fibroblasts and we put in the medium new fibroblasts which have not been irradiated, we observe an increase in the viability and the proliferation of fibroblasts. So there is a chemical activations of the products which have been produced previously from the other fibroblasts which were irradiated. So the idea now is that the component irradiated by the photobiomodulations, they goes in the extracellular matrix and they can travel into the tissue and reach other cells which have been activated in this way in a pure chemical way. So that explains the fact that despite the blue light remains on the surface, the product of photobiomodulation, they go deeper, and the healing process can go deeper. Now, let's talk about a few examples of red and blue and near-infrared photobiomodulation therapy experiments. First of all, let's concentrate on preclinical trials. Here we have an example of a wound created by a burn on a mouse, and we are treating, those people are treating with 810 nanometer, so we are in the near infrared, and effectively it works. After 9 days, the effects of photobiomodulation in terms of healing process is clearly visible with respect to the control. In this second experiment, always on mice, the authors are acting with the red and near infrared. The healing is created by radiotherapy, and effectively after a few 42 days about here, we can really observe a healing process due to the red and the near infrared. But even more interesting, we see that the red is more effective than the near infrared, which is very consistent with the theory and the simulation that we have developed before. This is an example in which we are using 420 nanometers, so blue. This is mice. The healing has been produced by a scratch, okay? And then we treated with 420 nanometer, and we observe with histology, so a gold standard, let me say, the structure of the tissue, treated and untreated. And we see that the structure of tissue in terms of distribution of cells, it's much more similar to the control than the untreated one. So the tissue in the healing process is really restructuring because of the photobiomodulation with the blue. And if we are analyzing the same tissues by using two photon microscope, we see that the structure of the cells, because here we have a cellular resolution, the structure of the cell and the structure of the collagen in the treated samples, in the treated tissues, it's much more similar to the control than untreated one. So that means that really wound healing at 420 nanometer, it's going to affect the morphology, the distribution, the cytoarchitecture, let me say, of the tissue and the structure of the collagen. Here you can see all the results resumed in terms of histology and in terms of healing. I mean, this is very visible on the skin how the healing process works with 420 nanometers. After a few weeks, we may observe a complete healing process. Now, if we move to clinical trial, we see that by using 420 nanometer, we observe a good healing process, okay, after a few weeks. This is a chronic venous ulcer. And always using blue light at 420 nanometer, we observe a good healing process also on venous ulcer and on post-traumatic skin lesions, which were chronic one, were not healing, so we unblock the healing process, and after 2-3 weeks, up to 4 weeks, we observe the complete healing process. Moving to the near infrared, also we have, these authors are working on leg diabetic, and they also observe a healing process after a few weeks by using 808 nanometer. So, to conclude, we can say that for sure photobiomodulation works. It works with red, it works with the near-infrared, it works with blue. We can say that the cytochrome C-oxidase is the main component which has been excited by the light and which produce all the component and the path signaling that is going to produce the healing process. But from our Monte Carlo simulation, we can demonstrate that the blue is from 30 to 100 times more effective in activating the cytochrome on the surface than the red and the near infrared because of the absorption capability of the cytochrome. And going deeper, but also by using Monte Carlo simulation, we can see that even if the red and the near infrared are going deeper, the cytochrome is absorbing much less. And so the capability to excite deeper the cytochrome with red and near infrared is from 100 to 300 times less than the blue on the surface. So we can say that blue excite much more cytochrome, but blue doesn't go deeper. How can we explain the fact that in clinics we see that blue works very well for healing? Well, we show that there is a memory effects. We show that effectively after the first process of photobiomodulation on the cytochrome, there is a release of the component of the photobiomodulation in the extracellular matrix. And these components, these biochemical components, are reaching other cells which were not irradiated and are chemically activating those cells. And with that, I would like to thank for your attention.